Joana, we are now continuing. We will continue now with EMODnet Biology, and it will be presented by the coordinator, Joana Beja, from FLIS in Flanders.
Yes. Yeah, there you go. Good afternoon, everyone. So I'm going to skip this slide because Connor has introduced EMODnet, so I don't really need to spend too much time with it. And I'll focus on EMODnet Biology, which is a consortium with 24 partners. On the right-hand side, you can see a bit of a map of a network. In blue, it's our partners. In pink, the links to other EMODnet thematic lots. And then in this brownish color, the links of the partners with the regional seq conventions, and on the black, it's other infrastructures or integrators. We do a number of things, like all of the EMODnet thematic lots, so we're not special in that sense. Publish data, create products, we provide services and tools for both users and providers. And one of the main remits is as well to support member states, EU institutions, and the regional seq conventions. This is a very high-level overview of the data flow within EMODnet Biology. So we have data providers within our consortium, but also data providers which are external. They can be from EU-funded projects. They can be citizen science activities, NGOs, researchers in your institutes, for example. And the data that we publish is also shared with the OBIS, Ocean Biodiversity Information System, and GBIF, which is the Global Biodiversity Information Facility. And it will be found within the EU DTO in Edito Infra when it goes live later this year. So what do we do? So we do a data publication. So we have a remit from the Commission. We focus on 9 sub-themes, which you can see from the images. And we deal with a lot of different types of data collection activities. It can be human observations, but also machine observations, imaging, tracking, acoustics, and we are now working towards integrating genetics. We do so by using and adhering to standards. So for the data on one side, and on the other side, you have the standards we adhere to for the products. So we use a standard that is widely used within the biodiversity community, both by OBIS and GBIF as well, which is the Darwin Core. We use the OBIS-ENF format, which allows for the link between the species occurrences or the taxa occurrences with environmental parameters or biotic measurements of the individuals. And we use vocabularies, controlled vocabularies. So we use worms for taxonomy, which has been spoken as well before in this conference. We use marine regions for locations, and the BODC NVS2 for any biotic or abiotic measurements, including sampling instruments and temperature, length, anything. And within the product side, because of centralization, we had to change our approach to how we publish our products. So we are now creating products in CFNet CDF format. It's not a straightforward job for biology as opposed to other disciplines, even though there is guidance, CF guidance on how the files need to be formatted. The inclusion of these files or the publication of these files in both GeoServer or ERDAP are not straightforward. So it's always a fight because the systems themselves, they don't really like them. They don't like taxa. So this is just a bit of a status overview of what we have at the moment. So we have more than 40 million occurrence records and 96 million extended measurements or facts. You can find them through the central portal GeoViewer and select the data that you want to then download. There are a number of filters which we will be expanding very soon. So at the moment, if you go to the viewer, you can only search by taxa, but we are in the process of adding other filters, so you can search by year, but also by data set, which could be quite relevant for you to show your management. We've submitted the data, there they are. And then in terms of the evolution, so the different colors represent the different phases. We are currently on phase 5, which ends in May 2025, so that's the darker blue. And you can see where we have the data gaps. So on one side it's a functional group, so we don't have a lot of data for angiosperms, mammals, or reptiles. And on the other side you have for marine regions. So Arctic, Mediterranean, and the Black Sea are the areas where we have the least amount of data. It's probably not very surprising for those of you working in marine biodiversity, and maybe within these regions, the constraints are quite well known. In terms of the data products themselves, so we currently have 38 products, but in the central portal viewer, we don't have them all on display. Some of them are quite old, so they remain in the catalog. As Connor said, they have CC BY licenses. We always aim as well to publish the procedures and the scripts required to create these products, and in a way that they are replicable, so you can use your own data sets and create a similar product with other data or for other regions. And then they are implemented in the portal, but with the adequate filters. Sometimes it's just taxa, sometimes we have time as well, so it very much depends. It's a varied approach. And on this one, I hope this works, because this is an animation. Yeah. It's not live. So this is the Calanus distribution that actually Charles developed in the previous phase. And you can see it's like model dated, gridded data, and you can see the abundance of Calanus through time. There is also an associated error layer. So this was done, I think, with the CPR data set as a base, right? Yeah. And this is just to showcase what we can do with the data that we have. I mean, as Charles said yesterday, even though we have 40 million occurrence records, we do have gaps in data. So we are limited in some areas on what we can do as well, and the type of output that we can create that is scientifically valid. So in terms of for external products, I just wanted to give a brief overview, because I know that some of you might be working with other EU-funded projects that have a requirement to make your outputs available to EMODnet. So this is a very high-level overview of what we do. But if you have an output from a particular project that could be, we could host, republish. We do follow some procedures. We do an assessment of how relevant that could be for the biology users. And then we assess how we can best implement it in the GeoViewer. It's not always easy. So we're currently dealing with a product that it's a very interesting one, but the projection is not friendly with GeoServer. So we had to request to the providers if they could reproject the data so that we can actually use it. So it can take some time. As you can see, it's really not... And we have our procedures that we use for our own consortium on how to create NetCDF biodiversity NetCDF files, which is open on GitHub, so you can also have a check and come back to us if you have any questions. And then lastly, I think I have 2 more slides. Just a brief overview of other things that we have, like tools and services, and where you can find them. So I believe the slides will be online, so you can then follow the links as well. But we have a course on how, I'm finishing. A course on how to reformat the data into Darwin Core, because that's the the format that we accept, the standard. And we have a number of tools, both for providers and for users. I will be presenting the QC tool tomorrow, so I'll just leave it there for you now. Yeah, thank you.
Okay. Any questions for Joana? Yeah.
Not you.
Thank you for the presentation. I have actually two questions. One is related to the data gaps that you were showing. So I was wondering if, for example, in the Mediterranean region, what we see is actually that data exists, but they are not shared yet. So this is, and so what you are doing to
we
disclose these data that are still.
Yeah, so we have some activities that, for example, in this phase, we had a workshop last year focused or targeting Mediterranean organizations, not just in Europe, but also on the southern side of the Mediterranean. But there were problems with visa applications, so a number of colleagues could not travel to the workshop. It was an in-presence only. It's also sometimes a political issue, so we can't overcome that in EMODnet. It's sometimes a cultural constraint as well, that data sharing culture is not the same across Europe, I think, as we all probably know. So it's targeted activities to going to conferences as well, presenting like Mediterranean focused or Black Sea focused. Yeah, it has to be like that, and trying to use the network for direct contacts, but it's not always successful.
Yeah, this was highlighted at the workshop at the beginning of the decade in the Mediterranean workshop, these gaps between northern countries and southern. The other questions is related to the submission of data products that you can. So is it a general, let's say, approach of EMODnet of embedding the products?
No.
Okay.
No.
And are you thinking of providing maybe requirements so it is easier for who is producing the products to be compliant with your...
Yes, so that link to GitHub already has some of the requirements, at least for the files, but we will be having a workshop later this year for the EU-funded projects, mainly, to explain a little bit more about the data and the data products, for example, so that we can work together and make it as simple as possible for the outputs to be integrated as and when needed. It's really not an EMODnet-wide approach, but we do understand in biology that we don't have the expertise across all of the sub-themes that we have to cater for. So, and it's better that we leave experts in a specific, example, in mammals or in fish, to do the work that is scientifically relevant than for us to try and make up something that the community is not going to use because they will not trust it, for example. So, yeah.
Good. Anybody else? Question? Maybe I have one question. You said that you're also going into genomics. Can you say a little bit more about that? What your plans are?
We can say a lot.
Oh, just a little.
So, GBIF has, I believe, a year ago, 2 years ago maybe, published some guidance on how to publish genomics data. OBIS has followed on that as well, and we are trying to adapt those guidance as well for EMODnet. It is within our remit for this phase to get a map with the distributions of the species or the taxa that are identified by genomics data. It's a lot of work because it requires technical development in the database, because genomics data doesn't work the same way as other types of data, as human observation data. The volume of data is also much bigger for genomics. So we're trying to integrate it, but it is complex.
What is the cooperation with EBI in that sense?
Well, yes. Well, EBI will have, as far as I know, the raw sequences. We're not going to store raw sequences. We're not storing that.
Could be a derived.
Yeah.
Okay, last question, because we have 5 minutes more, and then you.
Just a quick answer, because I'm with EBI, I'm with Magnify. We don't actually store raw sequences. It's mainly, we have a lot of different services, especially analysis pipelines. We can maybe kind of talk about this in more detail later. So I can just let you know what we can offer.
Good. Yeah. Cool, thank you.
Thanks,
Okay.